Good evening, everyone. Welcome to LumCon Science Talks. We have a very exciting talk for you again this evening. I hope you're all doing well and staying safe out there. My name is Mert, and so I will be your moderator for tonight. So to ask questions or get clarification on anything that our speaker brings up tonight, you can use the question box. I see that several of you are already giving me a hard time using the question box. But I did send a question, a food for thought question, which is, would you rather explore the ocean or outer space? And so if you can just practice using the question box by answering that question for me, we'll learn something new about you too. So remember, if you have anything that you want to communicate with either the speaker or with LumCon, please use that question box. I will now turn it over to our executive director, Dr. Craig McLean. That's my typo, by the way, in his name.
I still really like Crane McLean. Just I don't know, I like the alliteration.
Yeah. Please don't dock my pay.
Never. I think I'm going to keep it. Go with the name change. Well, it is my privilege tonight to welcome Dr. Peter Girguis, who's a professor of organismic and evolutionary biology at Harvard University and the co-director of the Harvard Microbial Sciences Initiative. And he received his bachelor's from UCLA, his PhD from UC Santa Barbara, and then was a Packard postdoctoral fellow at the Monterey Bay Aquarium Research Institute before joining Harvard. And so Professor Girguis studies how animals and microbes have evolved to thrive in their particular environments, and he's especially interested in the sort of animal microbial partnerships that occur at hydrothermal vents to the guts microbiomes of baleen whales. Among the many things that Dr. Girguis does, he also is known for developing novel open design instruments such as underwater mass spectrometers and microbial samplers for use underwater and potentially in exo oceans off-world. He tries to make these tools available to the broadest research community with the very important goal of democratizing science around the world. I was going to talk about all of the many awards and accolades that Peter has, but we would be here forever. He's the two-time chair of the National Deep Submergence Committee. He is a Gordon and Betty Moore Foundation investigator. He's won the Lindbergh Foundation Award for Science and Sustainability, among just the few things that goes to his accolades. Anyway, Dr. Girguis, thank you so much for joining us tonight. I really appreciate you joining us and very much looking forward to your talk.
Thank you, Craig. I am really grateful for those of you who are here tonight and for the invitation from Dr. Craig McLean and from LumCon and the like. I'm very sorry. I'm recovering from a bit of a nasty little cold. So my voice is a little scratchy. On occasion, I may have to mute and, a little bit of a cough, and then and I'll be back. I promise I'll try not to cough in your ears. But what a treat to be here. I have a real soft spot in my heart for LumCon. And in fact, Mert and I were just talking about my first visit to LumCon. I thought, wow, this is just like such a cool part of the world. And I think that there's so many cool things that are going on there that I'm glad that you all are taking advantage of that and here to join us tonight. Before I get started, I wanted to tell you a little bit about myself, because that may help you understand how I ended up, I guess, here doing all this stuff. I'm the child of immigrants, two immigrants who came from Egypt in the late 1960s. I grew up in Los Angeles and in a town called Downey. It was the aerospace capital, and there was a big aerospace company there that was the prime contractor, the major builder of the Apollo rockets and Skylab and the space shuttle. So I grew up in this really interesting town where we had people from all kinds of walks of life. We have blue collar folk, white collar folk, different cultures, different kind of creeds and religions, you name it. And everybody kind of worked together to make these machines lift off and do the exciting things they did. And I just thought this is how people lived. It rubbed off on me in a big way in that I love exploration and I love bringing people together from different walks of life because we all have something to offer. Everyone has got something interesting, some skill that they have that they can contribute for the betterment or for a better cause than just any one of us. So, you know, my original plan was to sort of stay maybe in California or maybe move to the South where my wife is from Tennessee. I ended up getting this job offer here at Harvard in 2005. I had no interest in taking it. I'd never lived in New England and I had no interest in doing it. And my wife encouraged me, said, look, you know, there are worse things. And if it doesn't work out, we can leave and it'll be just fine. I've been here since, and I love the opportunities I have to see and talk to people from all around the world and then share the kind of privilege and advantages that we have, bringing folks, again, from around the country and around the world together to work on new technologies to help us better understand this ocean. That's ocean that basically runs this planet. So what I want to do tonight is share with you some highlights of the kind of work we do, and equally important, kind of why we do it. And I will keep it hopefully a little bit on the short side because I'm excited to hear your questions. So thank you for your patience with me as I do that little preamble there, and let me get started on our show together here. So I will do, there we go. Excellent. All right. Well, I'll start with this title. This is not Planet Earth. I love this title, and it's not mine. Credit actually goes to Arthur C. Clarke, a famed science fiction writer, who said, you know, this is not Earth. This planet is ocean. And it's easy for us to forget that because we all live in places that are the green and brown parts, the continents. We're air-breathing animals that don't live in the ocean. But if you look at a picture of Earth from space, it's pretty clear that it's planet ocean. But it's probably more ocean than we even think. Now, the deep sea, which is the ocean that's below a half a mile down, kilometer down, that's 80% of our planet's living space. And that means that if you think of all the volume of Earth where anything can live, where an animal, a microbe can live, 80% of it is cold, dark, deep ocean. And so if aliens came to Earth, if aliens came to Earth and had to report back to their leader what the typical condition on Earth is like, they'd say it's cold, dark, wet, and salty, right? And this is, to me, it's just, it sort of reminds me again of what this planet we live on is really about. It's about the ocean and ocean life and how it supports those of us who live on land and all other habitats, including all of human civilization. So that means the rainforests, the deserts, you name it, Lumcon, Boston, wherever you may be, all of that is in this other 20%. Now, there we go. Let me find my little clicker there. So the one of the things that's really striking is just kind of how little we know about life in the ocean, relatively speaking. Now, you may hear scientists say this all the time, well, we know a little about this or we know too little about that. But at the end of the day, we really know surprisingly little about the ocean, especially the deep sea. And a big part of this, folks, is that we humans are not adapted to life in water. Pressures increase as we go down to the point where if I could transport any one of you to the bottom of the Challenger deep, the deepest point in Earth's ocean, and I were to measure the pressure bearing down on you, it would be about the same as putting 50 jumbo jets worth of pressure on your body. So that's a tremendous amount of pressure. Temperatures are near freezing. And, you know, honestly, many of the tools that we use haven't changed that much. Now, There have been big improvements, don't misunderstand me. But a lot of how we do ocean science is sort of baked a bit in some of the tradition and of how we've done ocean science. And it's important because it lets you make a comparison. If I go and I collect water with a particular kind of tool and I use it 20 years later, then that's comparing apples to apples. So sometimes, you know, we have to do that. But I would argue maybe one of the biggest challenges for understanding and exploring the ocean is that water is, now here's a nerdy word, electromagnetically opaque. What I mean is that all the stuff we're used to using through air, right? Radio waves, Wi-Fi, Bluetooth, GPS, cell signals, none of that works very far underwater. You can get some signals, go a little bit, but if you want to send a signal through the water from the surface to a half a mile down, you're stuck.
Hey, babe, can I have you come sign something?
You're stuck with really what I'll call like really low frequencies that don't really transmit a lot of data. So that's the real issue here, is that we can't just look down to the bottom of the ocean with our eyes. Let me give you another example. You can look up on a clear night and see the moon about 250,000 miles away or 380,000 kilometers away, and that you have no problem doing that. And our ocean, you know, at its deepest, it's like 11 miles, and you can't 11 kilometers, seven some odd miles. You can't do that. You can't even see a mile down, right? So that is a big difference between using a telescope to study the heavens and using, you know, a telescope to study seafloor. You're not going to get very far if you shine the brightest of bright lights. That is a major impediment. Now, because of that, a number of us have really tried to ask ourselves, how can we use machines to be our eyes and ears and noses in the deep sea? And so some of what I want to share with you today is about how we've built, you know, mechanical eyes and ears and noses for use in the deep sea. And I'm going to start by telling you a bit about these tools, and I want to tell you a bit about how you might have encountered them in different settings, or maybe a bit about how you might be able to build some of your own. And my hope then is to tell you a bit about some of the tools we've built, and then to turn around and tell you about some of the science we've done that tells us about the role the ocean plays in controlling our climate and some of the extraordinary animals and microbes that live in the deep sea. So, I'm a biologist by training, but one thing I want to say to all of you as members of the public, or maybe some of you are students, is that you have gifts that you can bring to bear on our study of the ocean, whether you are a marine scientist or not. Now, from a young age, I've been a tinkerer, and to tell you a quick funny story, in high school, my first car was a real junker. It was just a doozy. And it, you know, is what I could afford. But if I wanted to get anywhere, I had to fix it. And I was strongly motivated to go places. So I worked on that car day and night. I got a little shop manual, read about it, and I became very comfortable with assembling, you know, and disassembling parts of the engine. And believe it or not, that paid off in graduate school, where I wasn't afraid of using a screwdriver to take an instrument apart. As a biologist today, I believe I have an opportunity to put those skills and interests to use, because I know the kinds of questions biologists want to ask. And so we build tools to help answer those questions, right? And so we build what I call open design tools. So I want these tools to be broadly available to as many people as I can reach, who want to build their own. So I'm going to quickly tell you about some of these. I don't know if you can see my pointer, but we'll try. You know, we've developed a way of using osmosis, right? You know, this idea where salt sort of sucks water towards it. Some colleagues of mine actually 20 years ago, or a little bit more, came up with a really cool way of using osmosis to collect fluid samples in the deep sea. So think of it as like having an underwater sampler to collect little bits of water that you can study in the deep ocean without any battery power. That is a big deal because there are no electrical cords that just run all over the bottom of the ocean where you want to do your research. And we've adapted a lot of those for studying microbes. So we can take a water sample, analyze it, and do the kind of analysis to help us understand who's there and what they're doing. The other tools on here are an underwater mass spec. I'll tell you a bit about that. I'm going to skip over this one for today. And I'll tell you about a kind of our version of a Mars lander that I'm excited about. And I'll also tell you a bit about these microbial fuel cells, which I'm really excited to share with you. But we come up with other ways of collecting samples to preserve the materials in situ. That means kind of in place, so scientists can study them and understand how organisms make a living in the deep sea. And so again, I'm going to actually focus on this, and this lander over here. And let me start with the underwater mass spectrometer. Now, if any of you have been through an airport, you may have seen them take those little swabs and wipe them and looking for explosives, and that's a kind of mass spectrometer. Mass spectrometers are these amazing tools developed about 70 years ago that are so good at looking at different kinds of chemicals. Think of them as a kind of, to be a little nerdy again, if you watch Star Trek, as a kind of tricorder. Mass spectrometers are really good at sniffing a lot of different things and telling you what's what. Now, when I was a postdoc at the Monterey Bay Aquarium Research Institute, where Dr. McLean was. He was a postdoc there as well. And I had the real privilege of being supported by the president at the time, or CEO, her name was Dr. Marcia McNutt. I said to her, I've been working with these mass spectrometers in the laboratory, but the semiconductor industry, they're making a bunch of small components. And I wonder if we could take these little bitty tiny mass spectrometer components and put them in the deep sea and deploy a mass spec to the deep ocean. And I was, a measly postdoc. To Marcia's credit, as well as many of the scientists at MBARI there, they supported me in doing that and gave me a small budget to work with engineers to try a few things out. And lo and behold, we were able to take one of these detectors, a lot like what you see in the airport, and put it in a big, underwater pressure housing, right? So it wouldn't get wet, of course, and send it to the seafloor to sniff the gases that the microbes are using or making, right? And to sniff the kinds of different things that they eat and the things they produce. For those of you out there who are engineering minded, I'll just briefly say that the thing about mass specs is they require a high vacuum. So you have to use a big pump, in this case, a turbomolecular pump, to suck all the gas out, and then get a little bit of the gas in so you can actually analyze it. Now, how do you do that? Well, a lot of the mass spectrometers have a membrane, so they suck gas out of the air and bring them in, and then they can sniff that gas, right? So far, so good, except we're not in air, we're underwater, and I got 4,000 PSI on the outside, so how do you do that? And that was a bit of our secret sauce. We started thinking of how do we do this in a way that allows us to degas water. And what we found is that those thin little membranes are much stronger than you think if you give them the right kind of support. And so if anyone wants to ask about that at the end, I'll tell you about it. But we actually have a high vacuum on one side of this instrument and deep sea pressure on the other. And so we run water past that membrane, and it sucks the gas out of the water fast enough to analyze it in basically real time. So we can take this sniffer down, put it on a robot sub or a human occupied vehicle, and go around and start sniffing. Now, why would you want to do that? Let me give you this example. You are looking at a photograph of a hydrothermal vent, looking from the top down, and all this white stuff is microbes that eat sulfur, hydrogen sulfide, this toxic gas that could kill us. These microbes chomp on it, and that's how they make their living. We're able to take this mass spectrometer wand and slowly wave it over this whole site and start getting a chemical map that sits atop a real map. And we can start talking about which microbes are eating methane, which microbes are eating hydrogen sulfide, what microbes are doing what, and how much they're keeping it from getting into the overlying water. Now that is a really, really cool thing. And in fact, it's become something that's of interest to many other scientists. So there's about, I want to say maybe 9 or 10 of our underwater mass specs now around the world in different labs doing different things that are similar to what you see here. And it's important because it helps us understand not only how these organisms make a living, but how they shape the environment. This is an important point. Organisms shape the environment. Most of my work is in the deep sea, but some of our friends are using the mass spec in the upper ocean. And to tell you a fun story, excuse me, you may already know this, but about half the oxygen in our atmosphere came from marine microbes. And those microbes make a living by using all sorts of other compounds. And so my colleagues are using the mass spec to go sniff around the water, while others of us, myself included, are sniffing on the seafloor. So the mass spec's been a very powerful tool in that regard. Now I'm going to switch gears and tell you one of my favorite stories, and I hope you'll bear with me as I get through this, and then and share some details with you, and then we'll pick up the pace a bit. But I want to talk about these things that we call microbial fuel cells. Now, first I have to tell you a bit about some crazy microbes. So I have to forgive the schematics and words here, and if you don't know, you're not familiar with the terms, don't worry about it. I'm going to talk you through this. Basically, how do you stay alive? Well, the way you stay alive is you eat something, and then you use, or what it comes down to is the electrons in that, right? Electrons that are tied up in the food. You use that and oxygen to run kind of a tiny little power plant in your body inside your mitochondria. That's how you make a living. And in order to do that, you have to bring the food in and the oxygen in. There are parts of this world, though, including the deep sea floor, where if you go down just a little bit into the sediment or the mud, if you want to call it that, there's no oxygen at all. But microbes still need something to put those electrons on. There are microbes that have this ability to put electrons onto things that can do the job that oxygen does, but are solids. You may be thinking, okay, so what's the big deal? It turns out that rust is iron oxide, right? And rust actually is pretty good at accepting those electrons, but these microbes can't take rust into their body. It's a solid. So they've got this capacity to take the electrons in from food, just like you and me, run it through all those little tiny power plants, but then instead of sticking it on an oxygen inside the cell, which is what you and I do, they stick it onto rust outside the cell. Imagine if you sat down to your lunch and instead of taking a deep breath while you're, kind of chewing away and digesting your food, someone sucks all the oxygen out of the room and you're like, no problem, I'm going to stick my hand on this rusty wall and I'm going to breathe that. Or take me to the nearest railroad track, I'm going to breathe the rust on those railroads, beams there. Or find me a rusty plate, I just need to take a deep breath, right? That's what these microbes do. Crazy cool. And they do it because there's no oxygen in their environment, but there's plenty of rust and other minerals that are good at accepting the electrons, right? To do the job of the electrons. But you have to be able to get the electrons on the outside of your cell. You and I can't do that. No animal or plant in the world that we know of is capable of this. This is a special microbial thing. Now, this is where it gets fun. Over the last 20 years, my colleagues and I have been advancing this technology where we decide to trick those microbes. Instead of having them breathe away the rust that's in the sediment, we put a graphite bar or electrode and bury it in the sediment. Or we can drop it from a ship and have it plunge in. And the microbes grow on there and they start putting their electrons onto that graphite. Now, again, for those of you who are familiar with this, I can now connect that graphite with a wire to another graphite sitting up in the oxygenated water where there's plenty of oxygen. And you know that, if you see where I'm going, there's a difference in voltage between those two places that makes the electrons from those microbes go up towards this one. And that's an electrical current. And the moment we have that, and with some sophisticated electronics, we can use that electrical current to run sensors. And that's what we do. Now, so check this out. We want to study microbes that live in deep sea sediments. I want to figure out what they're eating, what they're doing, and all that. And we've built systems that actually capture electricity from those microbes to power the sensors that we use to study those microbes. It's amazingly cool. And we're able to collect and harness enough power over time that we can start talking between different sensors. Now, I mentioned that GPS and cell phones don't work, so we use acoustics. We basically shout through the water, but it's slow. But we don't need fast in this case. We're sending little bits of data, and we can transmit it over half a mile from one point to the next and start building a wireless seafloor network to better study the ocean without a whole bunch of expensive batteries or cables. Now, you may be thinking battery technology is getting cheaper. Well, this is true, but excuse me, it's not the cost of the battery exactly. It's really the cost of sending a ship and a robot sub or a human-occupied sub down to swap the battery. Even if you only have to do it once a year or once every 2 years, that's still probably going to set you back anywhere from $20,000 to $50,000 or $60,000. We put these things in and they run. And in principle, we've had some of these, in principle, they can run infinitely because they're an open system. We've set some up in some test beds, and they've been running, they ran for about over a decade before we just finally took them down. So, excuse me. So some of these technologies we're talking about leverage smaller consumer electronics that are being developed these days, technologies for other industries that are helping us better understand the ocean. And I'll end my little technology bit before I tell you about some science by introducing you to our version of a Mars lander. Now, one of the things that we do as oceanographers is we head out on ships a lot, and we'll dive down with a robot sub or a human-occupied sub, or sometimes we send robots out to go and explore. But it's interesting because when we send robots down, we'll often send them out with cameras and certain sensors, but we don't really have robots that are very good at measuring those chemicals, the kind of chemical sniffers or the noses. We've got robots, little robot torpedoes that have got good eyes, cameras, good ears, hydrophones and seismometers and stuff like that. But the noses, the chemical sensors, yeah, we got some. But we were inspired I personally have long been inspired by the landers we've sent to other planetary bodies. And not too long ago, NASA began to show interest in these ocean worlds. And I'm going to tell you a tiny bit about that at the end here. But there are moons of Jupiter and Saturn that have as much water or more water on them than our own planet. But NASA doesn't work in water. That is not what they do. They work in the vacuum of space. So when they heard about underwater mass spectrometers and chemical sensors, they were like, holy goodness, how do you do that? I, of course, was like, wow, how do you do that? But they funded myself, excuse me, and some colleagues to develop an autonomous observatory, our version of a Mars lander that we would send to the deep ocean floor, and to think about not just what tools do you put, but how do you run them? How do you run them? Now, if I have a certain amount of battery life, I cannot just turn on my fancy sensors and burn it in 12 hours if I need to make measurements over a month. So a lot of our focus has been on how do we use smaller low power sensors to do the sniffing, to figure out when something important has changed, and then turn on the big power sensors. And that's really what this lander is all about. For you technical minded folks, we've got the mass spectrometer on there. We have something called an isotope analyzer that helps us understand the origins of different gases. Temperature, we're measuring pH, which is acidity, measuring oxygen at a few different points on the lander. We can sample fluids and pour waters, and we can incubate them. We have high pressure vessels on board that we can take water samples in and then preserve them or do experiments. We were supposed to do this part. I haven't done it yet, but I wanted to put a manipulator on there so that we could tell the lander to, position a probe in the sediment or above the sediment. But remember, now we're talking, and if I've got, we got a whole bunch of onboard cameras, high def and 4K cameras, but you may be thinking, wait, Pete, you just told us you couldn't communicate data at broadband speed, so how do you do this? Well, I think the secret sauce to this entire lander project, let me hit pause real quick, has been this thing called an optical modem. Now, my colleagues at Woods Hole Oceanographic Institution and elsewhere have figured out how to use flashing LEDs or lasers to transmit data through water at the speed of light. Now, I was a little skeptical at first, but I saw it work firsthand about 10 years ago, where we were doing some work in the deep sea, and they proved to me that they were transmitting data over about, I think they said about just shy of 800, 900 meters, if I remember correctly. So call about half a mile, roughly speaking. Let's be generous, let's call it a quarter of a mile, right? And I was like, that's unbelievable. So you've got these two light sources flashing at each other, and these two eyes, these big light detectors looking, and they're moving data across it. Now, this is important, folks, because it's not only able to transmit data at broadband speeds, but now this is what we do with the lander, and I'm going to show you here. With this technology on the lander, I can drop this where I want to make my study, right? And then I don't need an expensive robot sub or human occupied sub at 30, 50, 60,000 a day. But if I go out with a ship and one of these optical modems on the end of a conducting cable, I just have to get to within 100 meters of this thing, right? Or about 300 feet, and turn them on, and they find each other. And I can now talk to it at incredible, at like broadband speeds, like from your home. And I can watch video, I can upload data, I can do all sorts of cool stuff, and I never have to actually physically touch it. This is a big, big advance, because every time we do work in the deep sea, if we need an ROV, that robot sub or a human occupied sub, it just gets expensive. It can, it just adds a lot of complexity. But to take a small ship out, lower this thing, and talk to this thing at broadband speeds is amazing. Now, I want to show you one of our first tests, because we rigged this at first to have a bunch of cameras and the optical modem. And I was like, what do I do for a test? And of course, we decided to do a selfie, because that's what people do. So what I wanted to do was to put this down on the seafloor with the optical modem, and then dive down with a robot sub, which we happen to have on board, because remember, this is a development thing. So I wanted just to have the robot sub there just in case this thing didn't talk and we had to bring it up. But I put the other flasher on the robot sub, and I said, okay, robot sub, let's get up right in front of this thing and look at it with our camera, turn it on, and tell it to take a picture or video of us, and then let's watch ourselves from the point of view of the lander. So that's what this little video is. So it's a little surreal once you see all this kind of happen. So we put this in the water, we let it go on its own, right? Release that, boom, down it goes. We then dive the robot sub, and we're looking at the lander here, and you'll see a blue light turn on.
Okay, it's talking back. Got a good light.
You heard my colleague there talk about that. Now this is us. This is the robot sub as seen from the lander. This is really weird. It doesn't even look real, but it was cool because we turned the lander cameras on, and we see ourselves, which is a very kind of close to a spiritual experience as you can get at the helm of a robot sub. And we see all these jellies swimming by, and it was just really cool to realize that we're able to see ourselves through the eyes of this thing that we're talking to at the speed of light. Okay, so that is really a lot of what we've been working on. And now I'm going to pick up the pace. I'm going to tell you some real quick science stories, and I'm going to turn it over to you for your questions. But what do we use all this stuff for? Well, the Abyss has all these sensors on board for studying methane. And methane is a really potent greenhouse gas. And much of the methane on Earth is actually tied up in the deep sea. Right? And it's important to know what's happening with methane, because again, as a potent greenhouse gas, it matters to the conditions on our planet. And in fact, some people even think methane played a role in previous mass extinctions, right? So that's not, it's not to be trifled with. Now, most of the methane is actually tied up in the seafloor. It's made by marine microbes, believe it or not. Most of the methane on Earth is made by marine microbes. And much of that methane bubbles up into the water and goes somewhere. We don't necessarily know where. We don't think there's as much leaving the ocean as we thought, we used to think. So somewhere it's being consumed. And a lot of research is focused on these kinds of microbes. Now, I'm not going to go into the details here, but there are two different kinds of microbes that form a partnership to eat methane. Now, I'm going to jump back here real quick and show you this picture. About 6 years ago, I was on an exploratory mission off the coast of Southern California where I grew up, off Point Dume, if any of you are familiar with it. And we did a dive just a few miles offshore, about maybe 5 miles, 8 kilometers or so offshore. And we're in about 800 meters of water, right, or about a couple thousand feet. And we find these bizarro areas of the seafloor which are releasing methane. Now, we've seen this before. They're called methane seeps. They often look carpeted, like that picture I showed you before of events, kind of white and yellow and orange and flat. It really looks like a 1970s shag carpet. But here we kept seeing these tall chimneys, and I'm going to show you another picture in a second, but they look like hobbit chimneys or little hobbit houses, bunches of them. So this is where we decided to do an abyss deployment. Like, what are these chimneys doing at the site, which clearly has a lot of methane? So what we decided to do was two things. Number one, we decided to recreate some of those conditions in the deep sea in the lab. And I just wanted to show you what some of our high pressure vessels look like so we can keep some of these microbes alive in here and pressurize them and study them. And then we decided to deploy the abyss. Now, up in the left-hand side, you'll see what I hope is a probably slightly jerky video, but this is 3D photogrammetry, and I hope it's working for you. It looks like it is. And it's basically a 3D model of one of the chimneys that we studied, taken using still images. But I want you to see what it looks like. It's very organic. This is about just shy of 4 feet tall, if I remember correctly. We see all sorts of evidence that methane is flowing through here, one shape or another. So we deploy the Abyss lander, and in the lower right-hand corner here, you can see the Abyss's probe stuck into one of the chimneys because we're sampling the fluids that we find therein. One of the things we then discovered is that there are microbes that eat methane that are found in the sediments around here, going to this lower right-hand picture, and microbes that eat methane in the chimney. And when we look at how much methane they eat by incubating them in the lab for a number of days, many days actually, almost a year, we can see how much methane they consume. Don't worry about what's on here. Just notice that we're at about 40,000. When we look at how much methane is being eaten by the microbes in the chimneys, it jumps up to the 400, 500, and 600,000 units, right? Again, don't worry about what it is, but this is crazy high. Almost every other environment we've measured on Earth is down here. So something about these methane chimneys is allowing them to maintain these really high methane oxidation rates. So to get a little into the weeds here, we started studying it with microscopy, and this picture shows you some of the rocks. See, this is 50 microns, so this is quite small. You can see some of the rocks here, and the bright spots are where we know there's cells. But then we're able to use different kinds of dyes, really, to light up the one partner I mentioned. Remember, I mentioned there's sort of two microbes in partnership. We can light up one partner in red and the other partner in green. And apologies now for those of you who have trouble with red and green, but just know that there's like these bright splashes of red and an awful lot of green and a lot of mixed red and green. But the point of all of this is that this chimney, without going into too many of the weeds here, it actually, as we look at these microbes, they form these really cool assemblages, these kind of like mats, laminations of microbes and microbes that we think are just passaging all the things they need to keep the methane consumption rates really, really high. And that was a really extraordinary finding, because now that we've seen these and studied them, and we kind of jogged our memory that of these little chimneys or chimlets, we start to see them in many, many other seep sites. And we're beginning to think that they're some of the most active places of methane consumption on our planet. And so these methane eating microbes are really helping keep our planet cool, or at least cooler, right? Because they're keeping some of the methane from escaping the ocean and into the upper atmosphere. What we don't know is what these microbes are sensitive to. Are they sensitive to a 0.5 degree change in the ocean temperature? Because they're going to experience that. What is changing the pH do to them, right? Does that mean more methane production, less methane consumption? We don't really know. So this is one thing that we're thinking a lot about addressing. Now, for my final story, I'm going to switch gears and just tell you a little bit about the amazing animals that live at deep sea hydrothermal vents, and then we'll wrap it up. Now, about 1979 or 1980, scientists dove on these hydrothermal vents in the deep sea for the first time. And it really changed the way we think about so many things. It was really in the 1960s that people finally had a model for plate tectonics. Before that, it was an idea that had been around, starting with a guy named Alfred Wegener and others who said, yeah, maybe the plates are moving, but no one had a mechanism. And then in the 1960s, some scientists said, oh, this could happen. But we really didn't have any firsthand evidence with our own eyes. We had a lot of circumstantial evidence. When they went and found these vents, areas where the seafloor was growing and spreading, that was like a smoking gun for plate tectonics. And geologists were thrilled. But the first people on board the cruises were geologists. And when they came back, they said, oh, it was amazing. These vents were amazing. Oh, but by the way, my dear biology colleague, yeah, there's some big animals down there. How big? Well, big. Well, what were they? I don't know, said the geologists. They're geologists. But when biologists eventually got their hands on some of these animals, they were blown away by what they saw. And this worm here, this red, fluffy looking worm, is the poster child for hydrothermal vent life. It's a worm called Riftia. It's all Riftia pachyptala, or just Riftia. But this is a worm that reaches about 3.5 or 4 feet long. The tubes can be about 6 feet long. And it is a worm with no mouth, no gut, no digestive tract the way you think about it, and no anus. And this really flummoxed biologists for years, and as they were trying to figure this out. And then they made an astonishing discovery that this worm has microbial symbionts deep inside its gut. And those symbionts convert carbon dioxide into sugars, just like plants do, using some of the same machinery that plants do. And that it's this symbiotic relationship between a worm that's given up the ability to eat, but instead takes up the chemicals from the hydrothermal vent water, hydrogen sulfide of all things, stuff that should kill it. It has this ability to take it up and safely take it to the microbes and do the same for oxygen. And the microbes use that. They take carbon dioxide to convert it to sugars, just like plants, to feed themselves and the microbe. Astonishing. The microbes that live inside here live inside host cells called bacteriocytes. I only bring that up because it's important to remember that these are microbes that have been almost incorporated by the worm, right? It is a key part of the worm. And in this figure here, this fuzzy red stuff at the top is its gill. This is how it takes up those chemicals that are needed. This thing is called its vestamentum, like a priest's vestament. And this is a muscular collar along with some hearts that hold the worm in its tube. And down here is where the symbionts live, deep inside the worm, literally billions and billions of symbionts. So a single adult worm has more microbes in it than there are people on Earth. Now, I'm going to just tell you that I'm really interested in studying how these organisms make a living. And to that end, excuse me, we bought a used 20-foot shipping container and turned it into a mobile high-pressure laboratory where we could recreate hydrothermal vents on board ship and keep worms alive in pressure vessels. Here you see little worm tubes inside our pressure vessels. These are their worm gills, their little red plumes, and they're just happy as can be, happy as a worm, happy as a clam, happy as a worm, whatever it is. But we do this so that we can recreate hydrothermal vent water in these columns you see on the left, these bubbly columns. We were basically bubbling sulfide and other stuff. These big old blue pumps back here, these are high pressure pumps. And these metal cylinders in the upper right, these are titanium pressure vessels that we can keep animals alive in. And we've got all sorts of analytical instruments here to make all sorts of chemical measurements. We got coffee to keep us going. And this is Dr. Roxanne Beinart, now a professor at University of Rhode Island. This is Dr. Jessica Mitchell. She's a postdoc in our lab. And we've done some really extraordinary experiments collaboratively on understanding how these worms work and what they do. In the interest of time, I'm going to skip over the data, but I want to show you one number here. These worms can feed their symbionts so quickly, and microbes are so fast at doing things, at least many microbes, not all of them, but many of them are so fast that they convert sugars so fast that this worm and its symbionts can add 10% of their body carbon a day. They can add about, it's anywhere from 5 to 10% of their body mass a day. So just imagine you go to bed weighing 150 pounds and you wake up weighing 165. So these worms are growing at extraordinary rates. And the reason is these hydrothermal vents are incredibly dynamic environments. And these worms grow fast, they live fast, they die young, and they grow like gangbusters. They make lots of eggs and lots of sperm, throw them into the water, hope for the best for their babies, and that's how they make a living. But our studies on these high pressure vessels allowed us to understand the biochemical processes that underlie these extraordinary observations. So until we actually built these pressure vessels, we had a lot of guesses as to what the worm was doing, how fast it would grow. We could collect them and study their biochemistry and understand that, but understanding their rate, their metabolic rates, required us to build these life support systems to keep them alive and actually study them at the conditions that they live in. So that's it for my science presentation, and I just want to wrap up by reminding us that understanding our own ocean is also a launch pad for understanding oceans in our solar system. These moons you see here, Enceladus and Europa, and Triton and Ganymede and Callisto and Titan, these are moons of Saturn and Jupiter, all of which have oceans, principally water, in some cases a methane ocean. But we are pretty darn convinced that there's liquid water, certainly on Enceladus and Europa, and that liquid water is covered by an ice sheet, just kind of like we see in the Arctic. And water, folks, is not limited in the universe. There's a quasar, that we know of that has 140 trillion times more water than on Earth. But we don't know what's happening on these moons, and there's a lot of possibility, a lot of interest in understanding if microbes might be living on these ocean moons in our solar system here. And so new technologies are taking we humans on deeper voyages into inner space and outer space. And it's really adapting and getting inspiration from other industries to think of new and exciting ways to have robotic eyes and ears and noses in the ocean, places we can't just take a stroll to. Like I said, I've been on plenty of 7-mile walks or hikes. And if I could walk straight down to the Challenger Deep, it'd be 7 miles, but we can't do that. So we need to use these technologies to really further our understanding. And I do really believe that crosstalk among different groups of people is the secret. So, if you're a software engineer and you spend your days developing, cell phone apps, and you really love and care about the ocean, seek out opportunities on how you might apply those, even just an hour here or there. If you're a student and you're thinking, oh, I love the ocean, but I don't want to be a marine biologist, don't be a marine biologist. If you're thinking, oh, I want to be an attorney, be an attorney. Humankind has evolved around the ocean. We originated, we are a part of the ocean. It is as much as it is a part of us. And so with that, I hope that you're inspired to think of ways that you can be a part of understanding our ocean. I also want to take a moment to thank the members of my lab. We have a lab tradition where each year we do a holiday card. And this year I found a wonderful cartoonist who drew the entire lab as Muppets. So you'll see their photos above and below and our Muppet selves here. And I really want to thank you all for your time. And I would love to answer any questions you have. Thank you.
Thank you, Peter. That was great. And we do have some questions rolling in. So I'm just going to give people a few minutes to do that. So we'll let them, and I'll remind the audience, remember, any of your questions can be put into that question box, and I will make sure to relay as many as I can as quickly as we can. So the first one is coming from Karen, and she's really interested in the worms. Her question is, once you take the worms out of the deep sea, are they sessile in captivity, or are they just free floating in those chambers?
Oh, that's a great question. That was Karen. Thank you for that. Mm-hmm. When we collect them from the deep sea, we have to use a robotic arm first and foremost. So we have to kind of smack their plumes around, unfortunately, but get them to pull into their tubes, grab them from the top, and pluck them like a bouquet of flowers. At that point, we put them in a box at the front of the submarine or the robot sub and close them up and bring them up. And as quickly as we can, we put them in ice cold water. And as quickly as I can, I put them in the pressure vessels, and they're just flippity-floppeting around. But I'll often rubber band little weights to them so that their bottoms are sitting downward, and we point the tubes up. Our aquaria are long and skinny deliberately to help them stay propped up. So that's exactly how it goes. And once we pressurize them, they're happy. And I've got lots of data that shows that they're just as happy in our pressure vessels as they are when we capture them. But we have a real strict timeline to follow to make that happen.
Great. And then kind of she says, wow, cool, thank you. Another question, follow-up question from Mike. Mike would like to know if the worms, if you changed the environment in the chambers just a little bit, could you get the worms to live longer in captivity than they do in the wild?
Ooh, I don't know. What a great question. This is my favorite thing about getting to share with you all, because you all have ideas that none of us have thought of. Two answers though come to mind. Two ideas come to mind. One is I have kept them alive in the lab. To my knowledge, I think we're probably still the only people who've been crazy enough to try this. But we brought them back to the lab at pressure, drove them from the ship right to the lab at UC Santa Barbara and kept them alive. We had them there for about 3 months. And then we had a big nasty storm that fouled our water quality, and then everybody died really quick. It was a sad day. That is the last time I've done that for that length of time. I really honestly don't know. I suspect that we may not be able to get them to live much longer than they do in nature. There are some worms that we know are about 3 to 4 years old, maybe about 5. And I would bet you that is about it. That's my guess, but I can't promise you that's true. So.
Awesome. And another question about the worms. Kirsten would like to know if in captivity they're increasing their body mass just as quickly.
Yeah. So thanks, Kirsten. That 10% of body carbon today is in captivity. Those are our measurements in our pressure vessels, which is astonishing. And was really just, we didn't believe it. And it took a student to join my lab and repeat my experiments independently to finally convince me it's true. So is that conservative relative to the deep sea? In other words, is it higher on the seafloor? I don't know. I don't know, honestly. And our best guess in the deep sea is that it's pretty darn close. And the way we concluded that, Kirsten, is by staining the worm tubes. And we can stain them, and then we can see new tube growth. And we can kind of estimate about how much carbon fixation that is. Does that make sense? But that's where we're at.
Alice needs a reminder about how long they live in nature.
How long do they live in nature? They live about probably up to 4 years is my guess. Maybe 5. That's really what, yeah.
Very short time lifespan for a worm.
Yeah.
Let's see, we have one from Peter. Peter would like to know, you were talking about the optical broadband. Peter is interested to know if there are concerns with the amount of light that's being produced with that technology, and if you foresee any environmental impacts.
Ooh, great question. Yeah, nice to meet you, Peter. I am not as concerned with the amount of light. And the reason is that there isn't sunlight below about 1,000 meters in the ocean, right? But there's plenty of light produced by organisms. And so a lot of them have well-developed eyes. And that is actually a reason for us to be careful about light pollution. The bigger issue is when we go down with an ROV or a submersible. I mean, we really bathe the environment in light. This is about as bright as, I would say, an LED flashlight, maybe a little bit brighter. So relative to the other kinds of light pollution we introduce, this is much less of a concern. So my guess is it's not a going issue relative to other kinds of things we do. So yeah, I'm not too worried about it.
And my understanding is the lights are not always on, right? They're just flashing when you're transferring information, and that's about it.
You beat me to the punch on that one. Yeah, so non-momentarily, and then we shut them down.
Excellent.
Good point.
Let's see. Oh, here's a good one, probably from one of our students. There's 3 questions related, so I'm just going to group everybody together. But everybody would like to know, with the emerging technologies for the deep sea, what careers do you foresee on the horizon that aren't necessarily popular right now, but might become popular and well-paying? I'll throw that in there because that was that was a caveat to one of them.
That is a fantastic question. I'm not going to tell you because I'm going to apply for that. No, I'm kidding. Look, so a couple of thoughts. There is a growing, I guess, awareness that we need to do a better job of proactively managing marine natural resources. Right? We've depleted something like 90% of some of the largest fishes in the ocean. I mean, it's insane what we've done. And this is too big of a topic in the last few minutes here, but I think there is a way to sustainably engage with the ocean. I think we have to do it. I believe a lot of that will rely on new technologies for understanding our impact and our contribution to species, to commercially viable, relevant species, to ecosystems and the like. I know that right now, even things like blockchain, which is a completely foreign concept to me, is being used in thinking about managing tuna fisheries, right? So I would encourage you all to think about what are the issues in ocean sustainability? Like, how do we do a better job of understanding and protecting the ocean, right? And come up with those questions. Separately, think about technologies that are being developed in other sectors that could be a sort of inroad into developing a new way for us to monitor and engage with the ocean. Adaptation from other industries is a very potent thing, and it is a good first start. And in terms of pay, I would just encourage you to really, be realistic about, earnings. But don't assume that, everybody's is completely impoverished. Our careers may not pay as much as those on Wall Street. There are other things besides salary that do matter, and that's up to you to decide what works for you. But there are people who are involved in the ocean sort of economy writ large who really do quite well, and others who maybe get paid less but have a heck of a wonderful life. So I hope that helps.
Absolutely. One question from Todd. Todd started off by saying he understands that we can learn about our other ocean worlds by through our ocean research here on Earth. But he's interested to find out if you think there are things that we can learn about our ocean by visiting other ocean worlds.
Yeah, that's a great question. I think, yes. When we look at Earth and we want to understand Earth, and I'm talking at a kind of systems level here, as a scientist, we have, like, how much replication can we do? Because there's kind of one Earth, right? Assuming we can't visit other dimensions, right? So we've got this one Earth. And that's what you got. So it's an N, as we scientists say, it's an N of one, it's sample of one. Understanding oceans and how they relate to other planetary bodies, for some questions, becomes an N of 2 or an N of 3. Now this enters the realm of how geology and geophysics interacts with an ocean that is well above my pay grade. But I can assure you that many of my colleagues are already thinking about how ocean chemistry is shaped by some of the geophysical processes on some of these moons, and how that might lead to different outcomes than Earth, where we have plate tectonics. That is not, there is likely no plate tectonics on Enceladus or Europa. What does an ocean look like without plate tectonics? And the first thought is that it's totally stratified. Like our ocean has a global conveyor belt that's driven by a whole bunch of things, but it is not likely to be as mixed as our ocean, and our ocean's already kind of stratified. So lots of really cool questions can come out of that, or answers, I should say, or insights.
Perfect. And I have one more, and it's kind of geared toward both of you. Crane McLean.
Crane, you want to go, Crane?
Crane is ready.
But there's been an interesting question posed to both of you. If you could visit anywhere on the seafloor, where would it be? Where's your next voyage going to go?
I think we should let you chime in with that, Mert, as well.
Oh.
I don't know, who wants to go first? I think, Mert, you've done the hard work. What do you think, Dr. McLean? Posting all this, you want to start?
I think my white whale of deep sea sites still has to be the Pacific Bear Zone. So this region of the South Pacific that is far, it's like right smack dab in the middle of the South Pacific, and it's far from land, so it doesn't get any food from land like the Mississippi might deliver into the Gulf of Mexico. It's underneath a very part of the ocean that doesn't have a lot of phytoplankton production, so there's not a lot of material raining down from the sea surface to the sea floor. It's far away from hydrothermal vents and other sort of tectonic activity that might produce more food. So it's really like this potential ocean desert, but previous researchers have been there and found microbial life. So in the absence of food availability, there's still microbes there. And so the question is then, okay, beyond microbes, when we're talking about like animal life, what might live there? What might be able to survive in this really different kind of place? And I think, if we could go there and explore, we might find fundamentally different and new kinds of organisms than we've seen in other parts of the deep sea, and might uncover, new biological processes and things like that. I just think it's a really fascinating place, and I think it's worthy of doing a lot more exploration of.
Mert, did you want to chime in too?
Well, given that I've never been to the deep sea, either remotely or in a manned submersible,
Yeah.
I do have to say that growing up in the Great Lakes region, shipwrecks hold
There you go.
kind of a relevance for me. So perhaps visiting and paying respects to a shipwreck
That is a cool answer. Shipwrecks are
of
amazing.
some kind.
They're amazing. Yeah, so shipwrecks are high on my list. I think I would be drawn to almost the opposite environment as my friend Crane. I think I would want to go. I'm gonna make this stick, by the way, Craig.
Oh, great. Fantastic. Thank you both.
I think that hydrothermal vents in the Southern Ocean off Antarctica would be really exciting. And I'll just be candid, that's driven by the explorer in me, you know, to think about these parts of Earth that are as far away from humankind as possible is really interesting. To think about how could you visit that in a respectful manner and to try and understand how that world works. And there's a bunch of things about the Southern Ocean that are different than events I've been to in the Pacific and the Atlantic. So that would be my choice. So.
Okay, I have to ask one more from Theo because it's specifically for Ukraine. Sorry.
What is that about a habit is formed by repetition?
He actually put that in quotes, so I had to say it. So Theo wants to know if the part of the seafloor that you mentioned might be an analog to what we might expect to find on Europa, since it is away from tectonically active areas.
Yeah, so it's funny that that's mentioned. I had a paper that was out that just came out today, actually, with my PhD student and master's student, River and Granger, and a really great microbial biologist at LumCon, Marshall Bowles. And we sort of proposed a dozen sites that sort of we thought may mimic some of the conditions that are either known or predicted to be in some of these, you know, exo oceans, as we call them. And so some of them were places we thought, you know, like at the bottom of the Marianas Trench deserves more exploration because it is the highest known pressures, and a lot of these exo oceans are much deeper than even our own oceans. Some of them are hydrothermal vent sites because they're the coldest or the warmest or hottest rather hydrothermal vent sites. You know, including some of the hydrothermal vents in the Southern Ocean that Peter mentioned. So yeah, and the Pacific Bear Zone patch was actually one of the sites we proposed as one of these study sites that we should continuously do more work on because they represent perhaps some of the closest Earth analogs to what we might find on, you know, these sort of other oceans.
Perfect. For those of you who I didn't get to your questions, I will get them to Dr. Peter as quickly as I can for answers. And so there's just a couple I didn't get to, so I'll connect you with those people if you don't mind, Dr. Peter. And perfect. And
Mert, I have one final question for you.